Hello mentees, I'm Dr. YoungRae, a mentor in the biology section. I'm very happy to meet you on uncounted video lecture. First of all, I apologize for my husky voice due to unexpected illness a few days ago. Currently, everyone is struggling with the COVID-19. The most important thing to suppress COVID-19 infection is to increase the body's immunity. For this purpose, the substance of interest is vitamin D. In other words, taking vitamin D increases autoimmunity. So what I'm going to tell you today is the preparations of vitamin D2 from Shiitake mushroom and its inhibition of Alzheimer's disease in vitro. Okay, the background of this lecture. As you already understood, vitamin D has two main functions. Vitamin D plays a role in health response, which is the classical role of vitamin D. The second role is hormone actions. Both of these roles are important, but what we are interested in today is the anti-inflammatory actions of vitamin D's hormonal role. We'll take a look at these functions later. Now move to two types of vitamin D synthesis. Vitamin D3 is synthesized by sunlight in your skin, and it can be synthesized from cholesterol isolated from goat wool. The vitamin D is synthesized from ergosterol from mushrooms such as Shiitake mushrooms. Today, I'll use the Shiitake mushrooms to synthesize the vitamin D2. Okay, let's discuss this process. Okay, preparation of vitamin D2 from Shiitake mushroom fruit bodies is the first topic out of two topics. This study consists of two steps. First, we process Shiitake mushrooms. These are aging and the chopping, UV irradiation, heat treatment, the making powder, vitamin D analysis, and the final products. All of these processes are done in GMP facilities. Let's take more detail. First step is preparing the Shiitake mushroom fruit bodies for samples. We process organic Shiitake mushrooms. Select the uniform Shiitake mushrooms. Wash them. Measuring the moisture content. In this process, the moisture content of Shiitake mushrooms is important. It is usually good if it is over 90%. Next step is aging, usually aged at 15 degrees C for 24 hours. At this stage, the tissue is softened. The third step is chopping the softened tissue like so does here. At this step, we measure moisture content. Moisture content must be greater than 90%. Okay, fourth step is UV radiation. The chopped Shiitake mushroom samples were spread on the stainless steel plates, followed by UV radiation. Important factors at this stage are UV intensity and UV irradiation time. As shown in this figure, vitamin D production is in equilibrium with other substances produced. So UV intensity and UV irradiation time should be well adjusted. In this case, UV was irradiated on 200 grams of chopped samples for 30 minutes with 2000 microwatts a square centimeter of intensity. At this process, it must be heat treated. This step is to produce the maximum vitamin D2 content. We process 24 hours at 60 degrees C. Next step is making the heated samples into powders using mills. And then vitamin D2 content in the powder samples is analyzed by HPLC. We got more than 300 micrograms per gram. It is higher vitamin D2 content than other companies' products. Finally, it is commercialization step. Brown powders in here. The final product is sold as a functional food ingredient. And dietary supplements like this are manufactured using these brown powders and be in market like a GNC. Okay, I hope everyone understood how to produce vitamin D2 using shiitake mushrooms. Okay, so now move to anti-inflammatory effect of vitamin D2. Second topic is the suppression of A-beta induced microglial activation of BB2 cells by vitamin D2. This research might be a little bit difficult for you, but it will be helpful when you are reading and writing scientific research papers. Let me briefly explain the background of this study. Okay, this is microglial cells. Okay, the first function of microglial cells are stabilize nervous cells. But when these cells are activated by some kind of activators to give inflammatory reactions, they can damage nervous cells to cause Alzheimer's disease. Okay, this is damage. So in this study, mouse, okay, microglial BB2 cells were treated with A-beta to activate and then treated with vitamin D2. Then what happened? You may know. The microglial cell activation is reduced by vitamin D2, right? Why? Because vitamin D2 acts as anti-inflammatory action. This slide shows a brief method. Okay. This is the microglial BB2 cells from mouse. These cells were treated with vitamin D2 and then treated with the A-beta proteins. Okay, after BB2 cells were treated with vitamin D2 and A-beta proteins, the cell proliferation was measured by CCK8 assay. And the biomarkers related to inflammation reactions were measured by Western blotting or light assay. And finally, ROS production was measured by fluorescence intensity. Results. This slide shows the cell proliferation by A-beta and vitamin D2 when measured by CCK8 assay. When you treat high concentration of vitamin D2 and A-beta to BB2 cells, cell will be dying. So we have to find a non-toxic range of concentration of these two substances. For BB2 cells, vitamin 2 was not toxic up to 1 micromole in here. And A-beta at 20 micromoles in here. So we use the 1 micromole vitamin D2 and the 20 micromole A-beta concentrations for the following experiments. After BB2 cells were treated with 1 micromole vitamin D2 and 20 micromoles of A beta, inflammatory factors of INAS and COX-2 protein expressions were measured by Western blotting. The expression of these two enzymes were significantly reduced by vitamin D2. INAS in here, COX-2 here. This means vitamin 2 act as anti-inflammatory actions. This slide is for pro-inflammatory factor levels. After vitamin B2 cells were treated with 1 micromole vitamin B2 and 20 micromole A beta, pro-inflammatory factor of TNF-alpha, interleukin-1 beta, and interleukin-6 levels were measured by ELISA. As seen here, vitamin 2 greatly reduced TNF-alpha, interleukin-1 beta, and interleukin-6 as compared to the A beta treatment. This slide shows ROS production by vitamin B2. After BB2 cells were treated with 1 micromole vitamin D2 and 20 micromole A beta, ROS production was measured by flow cytometer. As seen in here, vitamin D treatments significantly reduced the ROS production as compared to A beta treatments. This slide shows NF-kappa B inactivation by vitamin D2. After BB2 cells were treated with 1 micromole vitamin D2 and 20 micromole A beta, NF-kappa B activation, which is a key transcription factor in inflammatory response, was measured by Western blot. NF-kappa B is present in the cytosol without phosphorylation. When it is phosphorylated, transferred to the nucleus. Let's look at the panel B in here. The phosphorylated 65 protein level in nucleus was significantly reduced by vitamin D treatment relative to A beta treatment. In cytosol, non-phosphorylated 65 proteins levels was elevated by vitamin D2 as compared to A beta treatment. So this means vitamin D2 inhibits NF-kappa B activation. Taken together, vitamin D2 inhibits the production of inflammatory factors by inhibiting the activation of BB2 cells. Vitamin D2 binds BB2 cells for inactivation, and these cells reduce ROS production and suppress the NF-kappa B activation. Here too. So leading to the reduction of gene expression leads to the inflammatory response such as INAS, COX-2, TNF-alpha, interleukin-1 beta, interleukin-6. Okay, this signal transduction is a mechanistic action of BB2 cells inactivation by vitamin D2. So finally, this study means that vitamin 2 might prevent and treat Alzheimer's disease in here. Through this presentation, we discussed the production of vitamin D2 from Shiitake mushrooms and its anti-inflammatory functions in BB2 cells. I hope that the knowledge obtained from this presentation will help you in your further study. Thank you and good luck.